we talked about lots of things last time, and we spent a lot of time talking about like this, not this. We did talk about this too, but we spent a lot of time on this particular model talking about the differences between the measurement components and the structure components. And even in the video that I finally got posted yesterday evening, I interjected like a part in there where I'm like, I think I understand in sort of going back and reviewing sort of the questions and responses. I think there was a confusion about the word measurement and then measured variables, or like, using the term measured, where the squares, I think, were pretty, everyone's pretty straightforward that these are measured variables. But then we talk about measurement, we're talking about like the factors. So one thing to keep in mind is that these don't exist, right? So we can't measure them directly. They don't. Can't just ask people. I mean, you can. How depressed are you on a scale of 1 to 10? You can do that, but it's not likely to be very effective, and it's not like they cover a whole range of the kinds of things that contribute to depression. So we do these sort of factor models, these factor analytic sort of approaches, try to define this in a better way, in a more all-encompassing way that looks at the whole. My advisor used to call it bandwidth, the whole sort of bandwidth of something you're trying to measure, because there's definitely different aspects. Depression's a complicated thing to measure. So when we talk about measurement, we're talking about how are we defining these latent variables? How are these latent variables being created? So the parts of the model that represent measurement are the things that connect the latent variables to measured variables. So they're these loading and the errors around these measured variables. So yes, these are measured variables, but how they are being used to identify this latent variable is the measurement part, is the factor analytic part. So when you think measurement, think factor analysis, right, or latent variable. The structure is all about prediction. So going from gender to depression isn't part of defining depression. We're not using gender as an indicator of depression. It's just predicting it. We think there's a gender difference on depression. This is also hypothesizing that negative parental influence, this other factor, this other latent variable, its own measurement component here. There are two measurement components, right? This depression factor and the negative parental influence factor. And we are, in this model, hypothesizing that negative parental influence, this factor, predicts this other factor. And that part is also structure. And then the only other remaining thing, I always talk about there's two things. Well, there's measurement and there's structure. Measurement, structure. There's also things that are unstructured. Sounds like it'd be the opposite of structured. But it just means that they almost are considered unhypothesized. Sort of odd. It means that instead of saying, I think that gender predicts neglect, that I think there's a gender difference in neglect, and there's a pathway there. Because one of the things about prediction is that you are assuming some kind of temporal precedent when you put a prediction there. So that's a fancy way of saying that something has to come before something else. There's an influence. So to put a prediction from gender to neglect, I'm saying I think gender influences neglect. That somehow, boys or girls or whatever your hypothesis they're being more neglected than the other gender. There's some. It's that way, right? So by putting a double-headed arrow in here, I'm actually deciding not to make that hypothesis. I'm saying I think that there's a relationship between gender and neglect. I don't really know why. I don't know if really gender is influencing that. There's some relationship there. That's considered an unstructured portion of this. Now I could have just let's say I get rid of gender for a second altogether. And I just have depression as a factor here with these indicators. And I have negative parental influence there with those indicators. If I chose to put a double-headed arrow here instead of a single-headed arrow, I would lose this D because there's no longer any prediction. So it would just be two correlated factors. And without gender in the model, so pretend like that doesn't exist, that would mean that this is simply a confirmatory factor analysis. And that would be analogous to doing an EFA with an oblique rotation. Right? So I'm doing an EFA where these load on depression, those load on negative parental influence, and the factors are correlated. So that would be then a measurement model with no structure. I'm not hypothesizing a prediction between the factors. I just think they're related, and I want to establish that that fits my data. That would be a confirmatory factor analysis if this were double-headed and gender wasn't in the model at all. And that'd be like a two-factor, obliquely rotated exploratory factor analysis if we had done it that way. Does that make sense? Does that make measurement versus structure a little clearer? Is everyone sort of clear on those two differences? So everyone's cool. You got structure, measurement. So that was the thing I think that we sort of, going back through the video, I could tell there was some struggle to sort of understand what those things were and how measured variable is something different than measurement. So the components of the model. About recursive, non-recursive, it's all clear. It took me a while when I was first learning this to even, again, my brain kept wanting to remember them in reverse. Because recursive sounded like something that would have a feedback loop in or something. And non-recursive sounds like it isn't. So I kept remembering them backwards. Maybe that was just me. Maybe for some reason I had a problem with those terms. I don't know. Questions about those things? We didn't spend as much time on this. This seemed to be pretty straightforward, I think. And this will make more sense as well when we get to talking about model specification and identification. You'll quickly start to see as you start to run models, if you do it wrong, you'll get error messages saying cannot estimate the model. And oftentimes you might have some kind of feedback loop in or something like that will cause that. We talked about these different questions, all this crazy stuff like latent class analysis. I should mention too, like I mentioned it, we're not really going to talk about multi-level structural equation models, except with one special case, which is the latent growth analysis, which technically is sort of part of or a form of multi-level model that we'll talk about later on. But we're not going to talk about mixture or latent class stuff. We're not going to have time to go into all these very specialized types of models. Like even we're not going to talk about discrete time survival mixture analysis. We will focus on multi-group. We will talk about growth. But I mentioned all these here not because we're going to talk about them and to make you feel like, oh, why you mentioned these if we're not going to cover them. It's so that you plant these sort of seeds in your brain so you know these kinds of models exist and you can do them. And later on you can go sort of learn more about them sort of beyond this course. All right, so now we're the new stuff. So what are the limitations? There really isn't limitations in the sense of most people think of SEM as well, that's sort of on the next slide, where you're mostly exploring sort of correlational, non-causal sort of things, but that's because that's often how it's used. You can test experimental variables. You can do anything that you could essentially do with ANOVA, MANOVA. You can do those things in an SEM model and test them just the same. Just not often how it's used. The real limitation is that SEM is confirmatory. I mean, that may not sound like a limitation in the sense of the limitations of exploratory models is that you can't actually tell it what you want to test. It has to explore. So you have to have a model. You have to have something you're wanting to test. You have to have some idea of how the variables go together. In the case of factors, you have to know which items go on which factors. You have to have some hypotheses about how those factors relate. If it's a path model, you have to know the directions of how the prediction is going to go. You need to have some hypotheses about that. Now, that may not seem all that crazy because you have to have that if you're doing a multiple regression as well. You have to know what the DV is. You have to know what the IVs are. It's just can't start from scratch. You can't just say, look, I got a bunch of variables, tell me what things are related, right? So if that's where you're starting from, then start with an exploratory factor analysis. Perfectly fine. Go back to an EFA, do some exploration, play around, change the parameters of the model. Use a oblique rotation instead of an orthogonal one. Use different extraction methods like principal axis factoring versus maximum likelihood, or one of the other ones. There's like minimum residual and all these other different ones, and see what happens, see what you get. But once you have some idea of how those variables are going together and how they're working, and you want to test that or do some generalizability testing, see if it actually expands to more data, then you're switching over to confirmatory, and that's when you start. That's where you would begin to use a latent variable model approach, do something like structure equation modeling. So you need to have some theory. Cannot be used to explore possible relationships when you have more than a handful of variables. Exploratory method, so I say that it's confirmatory, but you have to start with a confirmatory model. You have to have some model in mind. But once you have that model and you test it, you can explore ways to make it better. So it's confirmatory, but there are these exploratory methods that can be used on top of your theory to improve your model. They're oftentimes called model modifications. So you can use model modification indices, which we'll talk about, to tell you what things can be added to your model or changed in your model to improve the model fit. SEM itself, remember, the data analyses are not what make or break the causality of a study. Doesn't matter how great analysis is using fancier stats. It doesn't make up for the fact that you have a crappy design or that you didn't do something experimentally. So SEM itself is not causal, right? It doesn't make things causal or not. Even though they are referred to as causal models, doesn't make it true. It's the experimental design that's actually going to make things be causal or not. And hopefully you spent enough time in like 692 covering like the different ways of validities and that kind of things to make things more causal or not. Those are the things that do that. And you can use structure equation models to test experimental designs, but it's the design, not the analysis, that make it causal. Just because this stuff is fancier than other things we might have used doesn't make up for the fact that how the data were collected is good or bad. SEM is often thought of as strictly correlational. I mentioned this a second ago, that we have a bunch of variables and we're trying to just test out the relationships between them. But it doesn't mean you can't use it with experimental data. You can test for things like mediation. You can look at manipulation, like experimental manipulation, to see if there's causal influence. Anything you can do with all the stuff we learned in 524, 485 can be done via a latent variable or a structure equation modeling lens. It's just that's not often how it's used. And that's because a lot of it is the social psychology field within psychology is one of the first to really fully embrace these kinds of models, and they would test all kinds of survey data and look at how things relate to one another, which gave it this association with correlational data, like field studies and non-experimental data. And they were trying to sort of make sense of how things related, and it sort of became almost synonymous with these kinds of large correlational studies. That doesn't mean that's how it has to be. So SEM is very fancy, does not make up for bad experimental design, which I just said a second ago. And data can only be generalized to the population at hand. So just because it's fancier doesn't mean it changes who you can generalize to and whether or not you can claim cause and effect in your design. Okay? The biggest limitation is sample size. So the more complicated, more complex an analysis becomes, the less powerful it is. We talked about that before, that when you go from a t-test to an ANOVA, you lose some power. T-tests are more powerful than ANOVAs because t-tests are more focused. You have one place to look, gives you the most power to find an effect that really exists. As you go to an ANOVA, that targeted difference that you have in a t-test now gets sort of diffused over multiple groups. So it's harder to find whether or not there are differences. You lose some power. When you go to multivariate designs like a MANOVA, you're now taking that diffusion you have in an ANOVA and spreading that over multiple DVs, which then, again, lessens the power. So as designs get less powerful, you have to have more subjects to make up for that and sort of bring it up to a power that you'd like it to be. So it needs to be large to set stable estimates of covariances and correlations. So one of the things I want you to keep in mind is, unlike MANOVA, ANOVA, the things we've sort of covered before this course, those things all did the analyses on the actual raw data. They would calculate mean, standard errors based on the actual scores. But the SEM, it's often referred to as covariance structure analysis because we're actually analyzing the variance covariance matrices, or the standardized version of that, which are the correlations. Because of that, our actual data, the data we're actually analyzing, is not the raw responses, it's the variance covariance matrix, which is why we can do things like replications that we're going to do as a project for the course, which is to take a correlation matrix or variance covariance matrix that is provided in a publication and actually put that variance covariance matrix into R and analyze that the way that the authors did in the paper. Because all we really need are the covariances or the correlations, depending on how they did it, what they provided. It's considered 200 subjects is if you have a small, fairly small to medium-sized complexity, meaning that you have a few to sort of moderate number of predictors and outcomes and parameters you're trying to estimate. And then 200 subjects is probably about right. And there's a study done now quite a while ago where they looked and said, well, the average sample size for most SEM models was about 200. That may have changed because that study was a little bit old. But the recommendation is something like a minimum of 10 subjects per estimated parameter, which gets tricky because we haven't talked about like how many parameters are we estimating in certain models. We'll get there soon. But the parameters that are usually estimated are things like error variances, the slopes, the loadings. As you go through and count up the number of variances that we're estimating, the number of covariances, the slopes, the loadings, count those up, multiply that by 10, and that's your minimum number. It's not the recommended number, that's the minimum. And it's a minimum to get you to a point where you can actually like mathematically compute things. So you're not going to get empty cells or things where you won't be able to invert matrices and do the mathematics behind the scenes. So it's also affected by effect size and the power of things. So the stronger things are related, the larger your slopes are, the larger your loadings are. You can find those with less subjects. So there's always a trade-off between that. But as models get larger and larger, more complicated, you need more and more subjects to sort of find everything and test the fit of the models and test all of the individual parameters for significance. You have to have enough subjects, and that's one of the biggest issues. It is a large sample analysis. You're not going to get by with very few subjects. When it comes to things like missing data, the issues, the solutions for it are the same as they are in things we talked about before. We talked about profile analysis or MANOVA. They're the same issues. The more DVs you have, the more things you have, the more likely you are that someone didn't respond to one of those items. Right? More variables means more chance of having something missing. In SPSS and the models, the analyses we've done so far, like MANOVA, for instance, or profile analysis, you had a couple choices. You can either go with the default in SPSS, which is to do a complete cases analysis, which means that anybody missing even one data point just gets left out. That can also reduce your power. It can also reduce your generalizability if there are certain types of people that are the ones that are missing. That's why we do things like a missing value analysis and look for data that is MCAR, missing completely at random, all those things we talked about before. Those all apply here as well. So you can go through your data, you can do the imputations using multiple imputation or expectation maximization, all these different approaches. You can do that before you bring the data over into an SEM model like Levon or EQS, M+, AMIS, all these different programs. You also have the option to just allow the SEM program to deal with it itself. So most SEM programs will be able to estimate the missing data, do an imputation, and run the model simultaneously. So as you're telling it you want to run a confirmatory factor analysis or a structural regression model, it'll actually go through, compute missing data based on the whatever information, there's different options. It'll estimate the missing data and run the model all together without you having to actually go through and separately impute data. So a lot of them have this built in, and you don't need to do anything. It'll actually just do it for you as built in as part of your analysis. Pretty nice. So I mentioned a second ago, the default in most everything, if you're dealing with SPSS, is just to do complete cases analysis, which is in SPSS referred to as a list-wise deletion. Many programs like SPSS and a lot of these SEM LVM programs allow you also to do pairwise, which means that it'll compute the correlations or the covariance and variances based on the data that are present. So it'll take the maximum number of people between two variables to compute the correlation, which means that each correlation or covariance may be based on a different number of people. Sort of odd. They'll have different n. So a lot of people don't go that route because of that, and they stick to sort of list-wise because it's easiest. And even when we did the multi-regression stuff in lab, there were times when we had to tell R to save the complete cases so that we can actually do some of the analysis. Okay, so when you're dealing with missing data, list-wise deletion is easy, but there's a lot of issues there. Like you spending time, energy, money on collecting data from people just to have them miss one question and then be left out of the analysis completely. That's not good. Seems like a waste. It also depends on sort of the percent of missing. Is there a pattern to the missingness? Because doing complete cases analysis assumes, just like imputation does, that data are missing completely at random. Well, this is missing at random, but usually it's termed as MCAR, missing completely at random. It means that the patterns of missing on any one variable, the patterns should be R. It means that the patterns of missing on any one variable are not related to another variable. So example of non-MCAR, measures of IQ and income. I think this is literally taken directly out of the 524 slides we talked about before, that measures of IQ and income subject below a certain level of IQ, like the cutoff for mental retardation or mental handicap, may not have any income because they're under guardian care, so they leave the income variable blank. So that those two variables, the responses to those two variables, the response to one variable is related to the missingness in the other, means that the data are not missing at random or missing completely at random, and there actually is dependency between the missing and the present of variables that we talked about in 524. So even the stuff, the imputation, the missing value approaches that are in these SEM packages or SEM software still require this idea of MCAR. You can't replace the data, impute them unless the data are missing completely at random. So it's still an issue, and then this is just an example. So complete case, right? So if you have 3 questions, the first person answers all 3, this person misses question 2, this person misses question 1. That means that this is the only person that will actually be retained when you do a complete case analysis, which is actually not good. You're missing dropping out a lot of people that have information that would be useful, but you're throwing it all away because they missed one question. So one way to get around this, and I mentioned this before, is called a missing value correlation matrix. It doesn't work that well for things like MANOVA or ANOVA because you actually are dealing with the means and stuff like that. Because we're going to be analyzing the correlations or the covariances, we can get by with pairwise deletion. For each correlation estimate, you are using the most data possible. Excuse me. Because you're actually going to be using the complete cases for each pair as opposed to across all the data that you have. You can have 10 variables, and it may be that variable 1 and 2 share, 90, let's say there's 100 people responding to 10 variables. 1 and 2 may share 98% of the variables, where there's 2, one missing from X1, one missing from X2. You can get 98 different scores for that correlation. But then when you go to 1 to 3, maybe they only share 95. Each pair of variables that you're correlating or computing the covariance for, you use the most amount of people you can for that pair for each correlation, which helps to preserve the most amount of information. But it means that each estimate is based on a different number of subjects. So the ends vary from across all the correlations, which can be odd, especially when computing degrees of freedom or other things you need to do. And there is a delete cases pairwise in SPSS, which works for things like correlations or other types of statistics, not so much for things like MANOVA or other things based on means and mean difference. And of course, there's imputation, which we talked about, some better than others, like variable mean insert. It's not so good. People like it. People will claim, oh, just replace the missing values with the mean, because it allows you to retain as many people as you can, and it doesn't affect the means because you're just replacing them with the means. The problem is, because you're replacing the mean for a bunch of people in the same variable, it does reduce the variance, because you're putting people in exactly the mean over and over again, which means their sum of squares, their contribution to the variance is 0. So you're adding degrees of freedom, but not adding to the numerator of the variance sort of equation, which actually could underestimate then the variance for those variables. Group mean insert is slightly better, that you look at the groups people belong to and you assign them the means for those groups. Not great. Regression is used. You take the variables that you have and you predict the missingness in other variables to replace them. It's better, again, than using the variable mean or the group mean. But it does, it's only really good if you use it sort of iteratively, which means you take some variables, you predict the missingness, then you use those variables to predict missingness in other variables, and you sort of keep doing that until the values that you're replacing are similar to the values that have already been estimated for it. So at the first time you use, say, variables 1 through 5 to predict missingness on variable 6. You then keep those missing in there. You use that, say 2 through 6 to predict values on 1. You sort of keep doing this until the values that you predict are the same as the values that were there in the model when you actually use them to predict other variables. That's what it means by sort of iteratively. So it keeps repeating until the values that you're putting in and out sort of stabilize. You get around the same number. There are better methods, like we talked before, expectation maximization, which gets used a lot, even in these SEM programs. We use an EM algorithm to sort of guess at the best values for imputing. Both of these, estimation, maximization, and multiple imputation, use some form of they generate values for missingness, and then they build in some randomness by randomly choosing then a value from a distribution of values to replace the missingness with, so it's not perfect. Using like regression and predicting a perfect score for that person and then replacing it. We're actually building in a little bit of random variation that exists in scores anyway, and using those distributions to then replace missing values so they're a little bit more natural and they sort of match with reality a little better. Multiple implementation, especially, it'll do multiple times or compute averages of the missing values that you want to replace. And this is nice because then it'll also give you a standard error for the estimate of the replacements. It'll tell you sort of how much those estimates sort of varied across different estimates from the multiple imputation approach. So you can actually then think about like a confidence interval. Here's the missing value with a confidence interval around it, so you can know sort of how much it varied or over repeated imputation. So what are the assumptions? Well, the reality is it depends on what kind of data you're dealing with. But if we're assuming a typical SEM where the data are normally distributed and continuous and all that stuff, then all the rules for MANOVA and those things all apply. They're all the same. We might make sure we don't have any univariate and multivariate outliers. SEM programs actually test for multivariate normality. There's something called Mardia's coefficient that we can look at in the output. It's basically a test for multivariate normality in addition to just looking at multivariate outliers. You can look for these things in SPSS or something first before pulling them into a program like EQS or M+ or Levon, which I actually suggest. Even though there are and some of these other programs, EQS, M+, they have functions for doing screening, for testing assumptions, just because of we're more used to working in SPSS and the functions in SPSS are a little bit easier to manipulate, to work around. All this sort of screening and stuff should be done before you bring it in to R or something to actually do these types of analyses. SEM programs, most of them anyway, that I know of, have corrected estimates. So they're oftentimes called robust methods. So if you suspect that your data is not normal, that it's skewed or you have outliers or something, and you don't want to get rid of them, you don't want you don't want to do some kind of transformation, you can choose to include robust estimates, which will then give you the regular estimates of this, like the slopes and standard errors and the fit statistics, and then it'll also give you versions that are corrected for the non-normality. So they'll give you these robust chi-square and slope and standard error estimates that take into account the degree of non-normality, how much you're violating, this multivariate normality assumption, and then corrects for it. It gives you estimates that are corrected and robust to those violations. It's also possible, something we're not going to spend a whole lot of time talking about, is to model goes beyond this. That if your stuff is not normal, it may be because of, if you have, say, like a dichotomous outcome or something that's only ordered polytomous, only has 3-4 choices, which obviously isn't going to be continuous. Or you have stuff that follows like a binomial distribution or some other type of distribution. These SEM programs actually have built into them ways of estimating models with those in mind. Just as for instance, if you have an outcome that's dichotomous, it's obviously not continuous, it's not normally distributed, it violates normality, all that stuff. Well, you can actually compute, instead of regular Pearson correlations, if you tell it that the data are categorical, it'll switch over to what are called tetrachoric or polychoric correlations. Fun words. So you have tetrachoric, polychoric correlation. This is when the outcome is dichotomous. This is when it is like ordered polytomous. What do I mean by ordered polytomous? assuming everyone knows what dichotomous means. So what do I mean by ordered polynomous? Right. So we have categorical data that are strictly categorical. There's no order. Something like marital status. All right. So you ask, or or ethnicity, where they're different categories, but there's not like an order to them. So for those we can even do those kinds of data, by the way, too. There are models for doing sort of nominal outcomes, but for doing corrected correlations, if you know that your data is dichotomous, you can compute tetrachoric correlations if they're ordered polynomous, meaning that you have something that's like rank ordered, or like many scales, they just use like 0, 1, 2, 3 as the responses. So they're not really enough to be a continuum, but they do have an order in terms of there's some moreness to it. As you go up in number, you're responding to more or less of some response that you have. And even to a certain degree, most of the stuff we use, like the Likert items, right, that go like 1 to 5, those are really ordered polynomous items, right? They're different choices, and yes, they're ordered, and there is some increase in there that's an ordinal variable, but there's not really enough to really consider it continuous. So sometimes correcting even for those, Scott Plunkett and I and a student, Ruby Jiang, that was here in the GE program a couple of years ago, we did a paper. We looked at a emotional intelligence measure that had been factor analyzed a bunch, and by factor analyzed, I actually mean they used principal components a bunch, never using confirmatory models, and never correcting for the fact that the responses were only, I think, 4 choices or something like that. So we reanalyzed everything using polychoric correlations, using some of these corrected estimates, and published a paper about it because it went, it contradicted some of the findings that people were using based on their principal components analysis. That was sort of crazy. It's like it had been like, let's say, 10 different publications, all of them used, and they were all sort of after one another, and they all used the same exploratory principal components analysis. They never even switched to like a factor analysis, and never did it confirmatory. It was really odd that it just kept repeating the same thing. It was very strange. So if you look it up, it's I want to say it's Jiang, Plunkett, and Ainsworth, but I could be wrong about the order. Where we actually, used a bunch of stuff. I think she did it in M+, but I could be wrong. It might have been Levon. The idea is that we can actually correct for the fact that even though we're expecting correlations that are based on normally distributed continuous data, we don't have that. We can actually compute correlations that correct for that fact going into the model, and then use corrected estimates for the slopes, standard errors, and fit indices that actually give you a better idea of what's really going on and how well the model really works with that kind of data. Barring what we just were talking about these corrections for, tetrachoric and polychoric and all those things, we're still typically assuming that especially the outcome measures are going to be continuous, normally distributed, which means relationships are going to be linear. So we don't want any multilinearity or singularity. And when we're actually testing our models versus our data, we want the residuals, right? The residual covariances, because we're going to try to reproduce it, and we're going to create a residual covariance matrix. If everything is working the way it's supposed to, all those residuals should be small. They should all center around 0, meaning that we're not getting any bias in the estimate. They should be fairly symmetric, which is why they're centering around 0. And if there's some asymmetry, then some covariances are being estimated better than others, and that's an issue. We need to sort of figure out why that's happening. That our model is doing a better job at estimating some things and not others could be problematic. So we are still assuming that this stuff should look, obviously this looks very different, but these are the same. This residual covariance matrix is very similar to in an EFA. We assume that if we're doing a factor analysis, that the residual correlation matrix should be small, and all these things sort of apply there too. But the no multilinearity and the linearity assumptions should sound familiar. It's all the same stuff we had in, say, MANOVA or in a exploratory factor analysis, all these other things we've already done. There's nothing new. It's all the same types of assumptions. It's just these are assumptions, yes, if you have these kind of data. The nice thing about SEM is that you can leave that and actually assume different things. You don't necessarily have to have linearity if you have, say, dichotomous outcomes and you're using tetrachord correlations and those kinds of things, okay? So all this stuff that we did before is just sort of a reminder. So what should you do to screen data before you're actually going to do an SEM? Well, you want to check for things like normality. You look for skewness or kurtosis. Should you do transformations? Well, it depends on how badly and what's going on with it. It's actually probably better to leave them as non-normal, like skewed, and use some of the corrections in the SEM software than it is to try to do transformation. That's something we can talk about later on. You want to test for linearity. Look at the data. Do they seem linearly related to one another? Do Pearson correlations actually make sense? That's what they're going to compute. They're going to compute the regular covariances that are precursors to Pearson correlations, which assume everything's sort of linearly related. If it looks like they're curvilinear related or there's other things going on, then you may want to switch the way in which the correlations or covariances are being estimated using some other methods, like we talked about, tetrachorics, polychorics. And just like with multiple regression, you're going to be putting predictors in the model together, predicting outcomes. And the same thing applies where things are too overly related. You have multilinearity, singularity. That's going to be a problem. It's going to make sure that the overlapping variance between predictors gets removed. Nothing can predict, and there's all these issues. So you want to make sure that the data aren't, I mean, obviously they have to be related, but you want your predictors to be related to your outcomes and your predictors related as minimally as possible to one another to sort of minimize the amount of overlapping variance that gets removed in predicting the outcome. So you want to make sure that there's no multilinearity or singularity for that, but also as you get closer to singularity, where there are variables that are almost like carbon copies of one another, they share, a ton of their variance. You actually likely you won't be able to invert the matrix anyway. The matrices won't be full rank. It means when you go to take an inverse, you got to find the determinant. You won't be able to, and that'll mean that that you won't be able to run the model in the first place. The biggest source of that, the biggest reason why that happens, is people oftentimes mistakenly put in the composite variable. They'll have, let's say, 5 items that are all measuring a scale, plus they have a composite, and they put all 6 of them in the model at the same time. So the composite is going to be highly related to all the items. They forget that they are related in that way, so that it causes some singularity, and the matrices won't invert. You actually can't do the mathematics behind the scenes because of that singularity. Right? You can't find a determinant, can't invert the matrix, can't go forward. Questions about the data screening stuff, about any of this stuff, measurement, structure, recursive, non-recursive, different types of models I talked about, like latent class analysis or mixture models and that kind of stuff, because we're not really going to talk about those again. I just wanted to plant those seeds in your brain so you know that they exist and you can actually go and find them. Has anybody ever heard of cluster analysis? Sound familiar? I have? Maybe. I don't know. I forget what I did yesterday, so it's very possible I could have mentioned it and then. So what's the difference between factor analysis and cluster analysis? I don't mean mathematically. I mean in terms of what it is you want to do. What's the purpose that's different? Because mathematically they're very same. They're very much the same. They use eigenvalue deconstruction. They use a lot of the same kinds of things. It's just the purpose is different. What are we doing with cluster analysis that's different than factor analysis? I should back up. What's the purpose of factor analysis? What is it you want to do? The main reason you do a factor analysis. I mean, and like an exploratory one. So you're trying to identify what variables go together to sort of identify a factor, right? So that's true. And that is like the theoretical sort of drive, right? We're trying to use factor analysis to support a theory that these items all go together and are measuring the same latent variable, right? But even in a more sort of, I don't know, meta view of that, the purpose is often just data reduction. We're trying to take a lot of data, we're trying to explain it with fewer things, and try to reduce things down. And we're doing that by grouping variables together, right? So we're trying to identify what variables go together and seem to be measuring the same thing, so we can sort of clump them together. We can actually put them together into a factor. So how then is cluster analysis different? It's still a data reduction approach, but it's doing it in a different way. What are we trying to do? Instead of organizing variables together into factors, what are we doing instead? It's grouping. It's actually not even trying to find factors at all. It's no longer, the point is not to try and find factors. We're not trying to group variables together anymore at all. What are we trying to group together? People. Exactly right. So instead of trying to cluster variables together into factors, we're trying to group people together into clusters. We're trying to look for types of people. So let's say you give someone a bunch of medical screening and you want to identify people who are high risk for heart disease, who are at medium risk for heart disease, and are at low risk for heart disease, depending on their responses. We're now looking at their responses to a number of variables and trying to cluster people into groups. So if you take a cluster analysis and you actually expand it out to a latent variable version of that, that's latent class analysis. The latent class analysis is the latent version, latent extension of a cluster analysis. Now they differ somewhat, but the goal is the same. We're trying to find out people that go together based on their responses. And that's similar to mixture analysis, mixture models as well, where we're trying to find different mixtures of people within a population, or that we are trying to group them too. The approach is a little bit different. Mixtures tend to go with things like latent growth models to look at different trajectories. They're different people changing differently over time, and that's the mixture. But they oftentimes are doing the same. There's even mixture models in SPSS that you can do to discover underlying subpopulations of people. But the idea, I'm just mentioning because I had mentioned latent class analysis, and that's the sort of latent version of a cluster analysis, much more like an EFA. There's like a k-means cluster analysis. It's like the one of the most common type, and so you're trying to identify people, which is oftentimes not what we are trying to do as experimenters. But if you're, say, a clinician and you're trying to figure out of people that you're working with, those who are at risk for, say, clinical depression or something like that, you know, cluster analysis may actually help you to identify those responses to things like depression inventories or clinical interviews that actually, where people tend to go together and respond in a similar way that indicates that they might have be at risk for clinical depression or all kinds of things. I mean, there's different ways it gets used. It's just you don't see it used in psychology quite as much because that's not often what we want to do as researchers, is identify the types of people. We usually already know the types of people that we're trying to work with and see how that differs on a number of outcomes. So it's just a reverse. We're taking outcomes and trying to find out who would respond that way, as opposed to identifying the who and seeing how they respond. Right? This sort of just a reverse way of doing it, I guess.